thank you. I don't know if there's any students here. I thought I saw a couple that were new. But anyway, my name's Mark Vanetta. I'm at the School of Public Health, Department of Epidemiology. I've worked at the Center for Clinical Trials and Evidence Synthesis since its start. I'm thinking about 20 years ago. And unfortunately for you all, maybe, or fortunately, I am going to be retiring at the end of the year. So you're going to get me for 3 months. So it's nice to have met you all, and I hope to give you a little bit more advice in the coming 3 months. So can we go through the students introducing themselves just shortly?
I can call people out. So if Jiajun would start us off.
Faculty.
Do you want me to start, or the faculty members?
Yeah, let's hear you.
Okay, so hi everyone. My name is Jiajun. I'm a SCD candidate in the clinical trials and evidence synthesis track. And in my last year, so this is the first time I'm going to present my overall 3 aims of my thesis dissertation. Yeah.
Great.
How about Riaz next?
Hi, I'm Riaz, 4th year PhD candidate track, and my research is in adverse events and clinical trials and systematic reviews, and yeah, I'll be presenting an RIP probably later in the year as well, because I'm hoping for some feedback on sort of how my aims are progressing. So yeah.
And then over to Alexandra Malinovska.
Hi, my name is Alexandra. I am a new master student joining you this year, and my first section here. I'm really looking forward and wishing Jiajun good luck.
Lynn Nan.
Oh, sure. Can you hear me, Taylor?
Yes.
Yeah.
Okay. My name is Lynn Nan. I'm a 2nd year SCM student with our track, and for my thesis, I'll be working with the vapors cohort to try to categorize their mental stress. And I'll be working with Dr. Janet Holbrook. Nice to see you all.
Jiangxing Ying.
Hi, I'm Xiang Ji. I'm a 1st year PhD student at the track. My research interest is methodologies of clinical trials and evidence synthesis. Good luck to Jiajun.
Well, you've come to the right place.
Other students who are tuned in that I didn't call? Okay, who's next, Mark? Faculty?
Yeah, just random. Yeah.
All right, Mark, you started us off, so over to Dave Shade.
I'm Dave Shade. I've been here in epidemiology for coming up on close to 20 years. Prior to that, I worked in the School of Medicine, still have a joint appointment there, and I work in coordinating centers of clinical trials.
Very good. Dr. Doug Jabs.
So I'm Douglas Jabs. I'm the director of the Center for Clinical Trials and Evidence Synthesis. I've actually only been here for about a year. And I chair clinical trials. I'm on my, I think, 10th trial. And they relate typically, well, I did a half dozen trials on the treatment of CMV retinitis in patients with AIDS, and I'm now doing trials on the treatment of uveitis and its complications. I've been working with the group here as my coordinating center, largely Janet Holbrook and Mark Vanetta, for almost 30 years. So I've been doing this for a little while.
Thanks. Over to Janet.
Hi, I'm Janet Holbrook, and I'm Jiajun's faculty advisor in the department and co-advising along with Caleb Alexander, who's also in Epi. And I'm like annoyed that everyone's outing me that I've been here so long, but yeah, it's been about 30 years.
Janet, you started when you were 2 years old.
Yeah, that was it. Thanks.
And Bobby next.
Sure. Bobby Scherer. I guess I'm a newcomer. I've only been here about 15 years. But I'm running coordinating trials and also been very much involved with the Cochrane collaboration while it was still here part of the CCTES.
Karen.
Hello, Karen Robinson. I am a graduate of the CCTS PhD in EPI, and I'm currently a professor in primary appointment in medicine, joint appointment in EPI, and I direct the evidence-based practice center here at Cochrane.
Stefan.
Hi, I'm Stefan Erhardt. I've been here just 8 years or so, so that maybe makes me the youngster. I'm faculty in epidemiology. I direct the track, and I work in multi-center as well as single-center clinical trials.
Other faculty tuned in that we haven't called yet? Mark, we have a couple guests tuned in. Shall they introduce themselves as well?
That'd be great.
Sure. Over to Alex McMahon.
Hello, I'm Alex McMahon, and I help support the CCT Center by performing all kinds of financial transactions. So I'm behind the scenes.
Okay, thank you, Alex. Over to Thomas Hardig.
Yes, hello, Thomas Hartung. I'm Chair for Evidence-Based Toxicology in Environmental Health and Engineering. But I'm also Professor of Pharmacology and Toxicology at the University of Constance and listening in for the first time.
Well, welcome.
Welcome.
And our friend Tony.
Hi, this is Tony Carlini. I'm a faculty member in Health Policy and Management, and I work with the Metric Coordinating Center, doing trials on orthopedic trauma.
Great. One more friend tuned in. This is Jiajun Jia. Would you introduce yourself, Jiajun?
Yeah, hi. My name is Yuan Xi. I'm a fifth-year PhD student in the track and advised by Dr. David Trentano and Stefan.
Great. I think that's all of our attendees for today. My name is Taylor. I'm the Research Program Manager to Dr. Jabs at the center, and I'm supporting the student RIP administratively today.
Great. So I think we can start our presentation. You can see the title on your screen. And
all right, I'll take over from here, Mark. Thank you.
Great.
Yeah, it's great to see so many new faces, old faces, and thank you for all coming. So this will be my first time presenting my doctoral thesis dissertation titled "Use Real-World Data to Understand the Use and Safety of Immune Checkpoint Inhibitors in Advanced Cancer Patients." So here's what you're going to expect from today's presentation. So I'll be going over background and aims, and then going to my methods and show some results and wrap up with a summary. So very traditional flow that you can expect from any kind of thesis defense presentation. So first of all, in terms of the why we are interested in immune checkpoint inhibitors. So first of all, we have very limited therapeutic options for late stage cancer. Here I have an example for non-small cell lung cancer treatment. So you can notice that in early stage cancer, basically you can do surgery, you have medications available, basically chemotherapies, and you also have radio therapies available to help you control the disease. But in the late stage, actually, there are 24% of patients that are not put on any kind of therapies. So the treatment is actually very limited for late stage patients. And also you might know that late stage cancer patients also have a very poor prognosis in terms of survival. So basically they're out of options. So immune checkpoint inhibitors are introduced recently, in the recent about 10 years. So it functions in a different mechanism. Basically, it triggers your immune system to kill the cancer cells rather than being toxic to cancer cells, and in the meantime also kill your normal cells. So there are 6 types of immunotherapy agents that are available by 2019. I list their names here, and also they're of different molecular targets. CTLA4 stands for cytotoxic T lymphocyte associated protein 4. PD-1 stands for programmed cell death 1, and PD-L1 stands for programmed cell death ligand 1. So the first agent that was on the market was ipilimumab, which is the CTLA4 inhibitor. And then in 2014, the first PD-1 inhibitor agent was introduced. And then in the following years, there were more PD-1 or PD-L1 related targeted antibodies. So in 2018, the first combination therapy was introduced. Basically, it was a combination of CTLA4 agent, which is ipilimumab, and one PD-1 agent, which is nivolumab. So most of the indication, most inhibitors target melanoma and non-small cell lung cancer, because those cancers are the most, in their late stages, they are the most, among the most poorly prognosed cancers. So they were tested very early, and then they were approved for those indications. So I would like to make an analogy of the immune checkpoint inhibitors. An analogy of immune checkpoint inhibitors using a car as an example. So imagine your immune system is actually a car. And normally the car runs on the highway and it targets your, the outside antigens. So the immune checkpoint inhibitor, the immune checkpoints actually serve as the car brake. So how cancer cells invade your immune system is that it actually pulls the brake of your car, and that way your car stops basically, and which means that your immune system cannot target your the cancer cells in your body. So what immune checkpoint inhibitors does is that it actually release your car brake again, and that way your immune system functions again, and it would target your cancer cells. So sometimes the inhibitors actually functions too well, and then it will basically accelerate your car, which means that your immune system can attack your normal cells. So that way we have very unique kind of adverse events, which is called immune-related adverse events. So they're unique in 4 ways. First of all, they may affect any organ. They may affect your skin to more important organs such as your lung, your heart. And secondly, the severity of the immune-related adverse events could vary. It could, you can have very mild symptoms such as fatigue, but it could also progress to very fatal cases such as life-threatening pneumonitis or myocarditis. And thirdly, the relationship with the response to the immunotherapy is unknown. There are a few reports that reported that theoretically, if a person has immune-related adverse events developing over the course of the treatment of the ICI, then the person is going to have a better response. But that's still controversial. And lastly, there's a delayed onset of the immune-related adverse events, which means that you might not have the adverse events when you take the drug, and all of a sudden, after a couple of months on the drug, you might start to develop those kind of immune-related adverse events. So how we study the immune-related adverse events is that usually the evidence pyramid is an iterative process. So after the drug is approved, the drug is approved based on the evidence that we generate from randomized trials. And the systematic reviews actually pick up the evidence from randomized trials and synthesize them in a way that it's appropriate to become clinical practice guidelines. So, and then physicians in the real world practice based on the clinical practice guidelines, and they also generate their own evidence by publishing potentially case reports and case series, or they have their own opinion on what works and what doesn't work. So how adverse events fit in this process is that they usually are, first of all, in clinical trials, we definitely monitor the adverse events. And then the evidence comes into the clinical trial, the clinical practice guidelines. But in real world, physician might encounter different scenarios where patients are not like the patients in clinical trials, and they might have a different presentation of the adverse events, so that they might also publish case reports and case series, or they follow up the patients and find that the adverse events are actually different in the real world than in the clinical trials. So how do we compare the evidence between or among these sources? So first of all, we noticed that patients are definitely more diverse in the real world. Here I have an example of melanoma patients in phase 3 trials for ipilimumab. So the patients are required to be adult patient, which means that they have to be aged over 18. And the other eligibility criteria include the patients cannot have any autoimmune diseases at baseline. The patients cannot be on any concomitant systemic therapies such as immunosuppressive therapies or any kind of systemic steroids. And they have to be in stage 3 or higher disease to be qualified for that trials, because those agents are really used in advanced cancer patients. And lastly, there are some requirements on the logistics as well. The patients have to have access to treatment centers with expertise to manage IRAEs. So interestingly, they found that they sort of divided patients in the real world. They have a real world database, and they divide patients into trial eligible with regard to trial eligibility. So they find that actually trial eligible and treated patients may have better overall survival compared to ineligible patients or eligible patients who are not treated in the real world. And another example is, so there are two, actually on the left-hand side, it's a real publication of a pivotal trial of immune checkpoint inhibitors. So they mentioned in their method that patients were ineligible if they had active autoimmune diseases or they have received systemic treatment over the past 2 years. However, on the left side, there's a study on observational database, and they find that more than 25% of patients with lung cancer or renal cancer had a comorbid autoimmune condition. So this result is quite surprising because those patients on the right-hand side might not even qualify for trial participation. So basically those patients are excluded, and you will never observe meaningful effects or effects of the treatment from clinical trials for those type of patients. So adding complexity to that, we have also seen there are different treatment paradigms in the real world. So based on the US evidence, what I can find is that there are two ASCO abstracts published very recently. One uses market scan commercial claims and encounters, and they find that there are 55% of immune checkpoint inhibitor prescriptions are from nivolumab, and also followed by ipilimumab and pembrolizumab. And they find that median out-of-pocket expenses is zero for those patients, because basically the commercial claims actually cover most of the treatment. Another study in the VA database, they've particularly in lung cancer patients, found that immune checkpoint inhibitor use for lung cancer is actually increasing. So there's no evidence on how the drugs are used in terms of switching patterns or adding on patterns, or if they are used in combination with each other. Globally, there's a survey among Chinese oncologists asking them, how you use those agents clinically. And they find that there's definitely increasing interest in prescribing those agents, but physicians are less educated on the mechanisms, as well as the immune-related adverse events management. And interestingly, they also find that there are over 70% of physicians who have ever prescribed those agents are off-label use. So there are a substantial proportion of off-label use. So it seems like the evidence are missing from the rest of the world, and I can explain a little bit about why. Because US and China are basically the two countries that have a lot of marketed products of immune-related and immune checkpoint inhibitors, and they're basically they're used a lot in clinical practice. So also relying on reporting of the IRAs in clinical trials are not always helpful, because the reporting of IRAs in trials of immune checkpoint inhibitors is not very good. There was one example conducted in 2015 looking at all the prospective clinical trials of immune checkpoint inhibitors over a 10-year span. And they found they used a 21-point quality score to help quantify the quality of report with regard to the requirement that's outlined by consort. So they find the overall quality is not good. And they also find that most of the issues happens in the definition of IRAs. Basically, the IRA definition are not clear. And also a lot of studies actually did not describe the time to occurrence, the time to resolution, and also there was no mentioning of the management. So putting all these together, and also when we consider decision-making on healthcare, it really should be based on the evidence of both benefit and harm. So how about we have a pool conducted or reported randomized trials, and then, so we start to question, are these systematic reviews about safety actually are reliable? And ultimately we want to know, if the clinical practice guidelines that are based on systematic reviews and clinical trials are reliable. So here we have, we sort of summarized in one sentence what the issue is. So basically it remains unclear if and how the effect in clinical trials can be translated in the real world, particularly in our case for patients treated in the real world with immune checkpoint inhibitors, and those patients are diagnosed with advanced stage cancer. So we identified 5 gaps. First, patients' characteristics might differ in RCTs versus in the real world. And secondly, treatment paradigms are more complex in the real world. And third, for patients treated in the real world, they might not qualify for RCTs at the beginning. And also, regarding the relative safety, RCTs are underpowered to compare IRAs between interventions. And lastly, there is literally no evidence on head-to-head comparisons between PD-1 or PD-L1 inhibitors because those agents are developed by competitor companies, and they won't compare their products to the products of another company. So the solutions that we propose is that we want to use real-world evidence to understand the use and safety of immune checkpoint inhibitors. So the real-world evidence are the data relating to patient health status or the delivery of healthcare routinely collected from a variety of sources. That's defined by the US FDA. So in our study, we're going to use a big administrative claims database, which I will introduce later. So the aims, for the aims, we're literally asking 3 questions. First, who are the users of ICIs and how are ICIs used in real world settings? And second, do the risks of immune-related adverse events differ between patients who would have met trial exclusion and patients who did not meet trial exclusion? And third, does PD-1 inhibitors cause excessive risk of IRAs compared to PD-L1 inhibitors in the real world, or vice versa? So to answer these 3 questions, we have 3 aims. The first aim is to sort of characterize the use of immune checkpoint inhibitors. And the second aim is to understand the difference in IRA risks between trial eligible users and ineligible users. And the third aim is to do a comparative safety study of immune checkpoint inhibitor therapies in the real world. So the data we used is called the Optum Labs Data Warehouse, and it has over 160 million de-identified commercial insurance or medical advantage plan enrollees. So that number is by 2019, I believe. And below, I have a graph that's in 2016, what's the representativeness of the database with regard to the overall US commercially insured or the US Medicare Advantage or the US Medicare Part D pharmacy claims. So we're only using the claims portion of the database, which means that we are not going to link the claims to the EHR because first of all, the overlap between the claims and the EHR are not very good. And secondly, because we're looking at, the information from the EHR itself is not optimal because most information are derived from natural language process. So it might, you might misclassify patients just based on the information that's partially from the EHR. So for the 3 aims, I have illustrated, the overall flow of how we define our population and how it relates to our target population. So we identified all the adult patients with at least one immune checkpoint inhibitor related claims during the time frame of 2011 to 2019. So 2011 is when the first agent was introduced. So we basically covered all the adult patients so far. So for AIM-1, we required the patients to have at least 12 months of continuous medical and pharmacy insurance in the database prior to ICI initiation. So the target population we have here is all the commercially insured ICI users in the US from 2011 to 2019. And second, for the second aim, we further restricted patients to have baseline lung cancer, melanoma, or urothelial carcinoma, or head and neck carcinoma diagnosis in the frame of 2016 to 2019. So why we picked that time frame is because there's no comparison between CTLA-4 inhibitors and PD-1 inhibitors before 2015 because PD-1 agents were not available until 2015. So to ensure comparable groups, so we have to limit our analysis to the most recent dates. So the target population for AIM-2 is commercially insured ICI users with those cancer diagnoses of interest, and also the time frame is 16 to 19. And for the third aim, we further restrict our population to PD-1 and PD-L1 users. So there are a couple of reasons. So first of all, CTLA-4 inhibitors are really primarily used in melanoma patients. So basically, you can't find meaningful comparisons between CTLA-4 patients and PD-L1 inhibitor users. And secondly, as I have mentioned before, PD-1 and PD-L1 inhibitor users are really, they did not exist until about 2016. So we sort of limited time frame to 17 and 19. And we restrict the indications to two indications, which is lung cancer and urothelial carcinoma. These are the major ones that are indicated for PD-1 and PD-L1. So the target population for this aim is all the commercially insured PD-1, PD-L1 users with lung cancer and urothelial carcinoma in the US. Actually, there's a typo. It should be 17 to 19. So the approaches that we took for AIM-1. So overall, the aim is to characterize the real-world use of immune checkpoint inhibitors. So we conducted a retrospective cohort study. We applied a new user design, which means that subjects enter the cohort when they are prescribed the new medication. So the index date is the earliest claim for any immune checkpoint inhibitor for each individual. So the patients are eligible if they are 18 years or older at the time of ICI initiation, and they have to have at least one claim between 2011 and December and 2019. And they have to have continuous insurance coverage for at least 12 months before the index date. Regarding the patient's characteristics, we assessed the characteristics at baseline, which means it's 12 months prior to the index date. And the information we collect was demographics and some clinical characteristics such as cancer at diagnosis, the type of ICI initiated, the comorbidities. We was focused on autoimmune diseases and brain metastasis. And also we tracked the history of chemotherapy as well as corticosteroids use. So as a measure of global function for these patients, we also predicted ECOG performance status. We used a published algorithm to predict these ECOG performance status and we sort of dichotomized it into 0 or 1. 0 basically means the ECOG is less than 2. 1 means that ECOG is bigger than 2. So ECOG of 2 is usually a cutoff for patients to participate in oncology trials, especially in ICI trials. Most studies exclude patients who have ECOG that's more than 2, which I will explain, which I will dive into details later. But here, just to get a sense of what ECOG does. So here's what the new user design looks like. So we have the index date determined by the first claim of ICI, and then we look back 12 months of continuous enrollment to look for information in for baseline assessment, and then we follow the patients until the outcome of interest or until the patient is censored. I will discuss the censoring criteria later. So we use it because it's sort of a gold standard in pharmacoepidemiologic studies, and it could adjust for confoundings at initiation, without bias during follow-up. Basically, it's gold standard and avoid selection biases. In the aim-1, remember that we want to characterize the use of immune checkpoint inhibitors, so we want to understand the treatment patterns. Here we have 4 types of treatment pattern. So first, we want, it's immune checkpoint inhibitor monotherapy, which means individuals complete a single immune checkpoint inhibitor during one continuous period. So if a person starts, here I have nivolumab as an example. So if a patient starts nivolumab on this person's index date, and we follow this person throughout the treatment course, if we don't discover anything else, then the person is on a single nivolumab treatment. And secondly, combined treatment, which means that, so if the patient starts both treatment on the same day, on the same index day, then this person is on a combined therapy. Or if the person start one of the treatment on the index date, and then over the course, the person has another treatment for over 75% of the whole more than 75% of the whole treatment course. So we define its overlap. So that way, we also define it as a combined therapy. And thirdly, for add-on therapy, very similar mechanism, but we require the overlap to be less than 75%. Maybe the person had nivolumab for 10 months, and then for, within the 10 months, maybe there's 1 or 2 months that the person is put on another treatment, such as ipilimumab, then we consider it's an add-on treatment rather than a combined therapy. Lastly, if the we also define switch patients, which means that if the two segments of ICIs are not overlapped, then we consider them to be switch patients. And I also want to discuss how we define our trial eligibility criteria. So first of all, we basically did a review on all the published, not published, but all the ICI pivotal trials. And we define ICI pivotal trials as those that are cited in the drug label. And then there are at least 1 immune checkpoint inhibitors in one of the arms, or one of the intervention arm. And also the trial has to be completed by December 2019 so that we have meaningful data out of the trial. So we also complement the trial information from the label with information from the FDA medical reviews, the information from clinicaltrials.gov, and also peer-reviewed literature. And with all the information, we try to find the eligibility criteria that are common, that can be applied applied across different cancer types and that are not related to concomitant therapies, and also reflective of individuals' global function. So across, so we find 26 trials that meet the criteria, and all trials require patients to be free of recent immunosuppressive agent use, autoimmune diseases, and untreated brain metastasis. And also the requirement for ECOG performance status are greater than 2. That way they're not eligible for the trials. So at the end of the day, we come up with our hypothetical exclusion criteria, which is ECOG score greater or equal to 2. And if there are any autoimmune diseases at baseline, and if the use of corticosteroids exceeding the daily hydrocortisone equivalent dose of 40 milligrams within 14 days before the index date, or there's a recent brain metastasis diagnosis within 28 days before the index date. So some baseline characteristics for AIM-1. So basically we look at 3 types of initiation. So CTLA-4 monotherapy initiation, PD-L1 patients, inhibitor users, or the combined CTLA-4 and PD-1 inhibitor users. So altogether we have 18,000 patients, and there are some difference in the age, and obviously there are some difference in the distribution of sex and race. But here the meaningful results is that before 2015, you literally see no users of PD-1 and PD-L1 inhibitors. So that's why we sort of limited our population in our AIM-2 and AIM-3. And some more characteristics. So what's meaningful here is that CTLA-4 inhibitors are most likely to be used in melanoma exclusively almost. And then you see more diverse population of PD-L1 inhibitor users because they're indicated for multiple cancers. And then the patients have different baseline comorbidity score, and they are also different in terms of the distribution of comorbidities, which I did not show here due to space constraints. Basically, there are differences in terms of a brain metastasis proportion, and also the predicted ECOG scores are different across groups. But you can see here, there are a good chunk of patients who have ECOG scores that are greater than 2, which means that they would not have been qualified for any trials. And we also examined the previous chemotherapy experience. It turns out that there are a substantial number of patients who do not have previous chemotherapy, which means they're first-line immunotherapy users. And also there are some, about 6 to 7% of patients who have recent corticosteroids use at baseline. So regarding treatment patterns, we have shown here. So basically, if a patient started PD-L1 therapy, PD-L1 inhibitor therapy at the beginning, then this person is not likely to switch to other kind of treatment. But however, you can see if the patient actually started CTLA-4 inhibitors, then the person is very likely to switch to another, PD-L1 inhibitors or add PD-L1 inhibitors during their treatment course. So you might question, so Jiajun, you mentioned before, there's a tendency that PD-L1 inhibitors are used recently. So I have to mention that this table is for patients get treated after 2016. So there's a, so the comparison groups are comparable in terms of, it's not influenced by the availability of the treatment. And also in this sentence actually gives you a sense of how the treatment are changed over time. So basically, I also did an analysis for patients before 2016, and I also find that there are 4.5 and 18.2 patients who started CTLA-4 inhibitors actually eventually switched to or add on PD-L1 inhibitors. So the trend is constant over time. So I would say the CTLA-4 inhibitors actually have a higher proportion of switching or add-on treatment during their course. So for the four defined hypothetical exclusion criteria, basically ECOG score, autoimmune disease, recent brain metastasis, and also recent corticosteroids use, you notice that there are, so across the board, if you look at the last column, there are a substantial proportion of patients who meet at least exclusion criteria, which means that they might not be eligible for clinical trials just based on one single criteria. But among these patients, the urothelial cancer patients treated with PD-L1 actually have the lowest proportion of meeting exclusion criteria. However, for melanoma patients treated with the combined therapy, the proportion of meeting one of the exclusion criteria is the highest. So the key findings for AIM-1 is that of 18,000 adult patients, most of them are PD-L1 users, and also only 2% of PD-L1 users switched or added treatment during their treatment course, whereas over 20% of patients who started with ipilimumab, which is the CTLA-4 inhibitors, actually added or switched to PD-L1 inhibitors during their treatment course. And also for 14,000 patients that are treated for approved indications, over 50% would have met at least one of the four hypothetical exclusion criteria. And among them, the melanoma patients treated with the combined therapy actually had the highest proportion of meeting the exclusion criteria. Lastly, trial participants were less racially diverse and younger than real-world patients. And among the 26 ICI pivotal trials that are pulled from existing literature and information from the labels, in 7 out of the 26 trials, white patient participants actually consist of more than 95% of total randomized participants. Whereas in our observational study, we noticed that there's only 75% of patients that were white. And the population in the real world is more diverse than those in clinical trials. So for AM2, we're trying to compare the risk of IRAEs by trial eligibility. Again, we used a retrospective design with a new user design. And here, the follow-up criteria is that we follow up the patients until the first occurrence of the outcome of interest, which is IRAE in this case. And they are censored at death, disenrollment from healthcare plan, or the end of study period, which is December 31st, 2019. So the eligibility is pretty much the same. They have to have ICI claims and over 18 years old, and they have to be diagnosed with the 4 cancers of interest, and then they have to have continuous enrollment for 12 months before the index date. So we have the trial exclusion status that is a dichotomized variable based on 4 criteria that we used before. And also the outcomes are immune-related adverse events. They're identified by the ICD codes in the claims. And we only document the first occurrence of IRAEs. So we constructed the cumulative incidence of experiencing any IRAs or specific IRAs. We also conducted Cox proportional hazard models to see the hazard ratio of experiencing any IRA. So the approach that we use are cumulative risk curve by exposure status, as well as the Cox proportional hazard model stratified by cancer types, because we allow there's a different baseline hazard for different cancers. So the cumulative incidence of IRAs across across patients, regardless of the trial eligibility criteria, is that basically over 60% of patients would experience at least one IRA at 12 months. And also, I listed some of the IRAs that are of very high cumulative incidence, which means that they're at least 15% at 12 months. So you get a sense of what kind of IRAs are expected for those patients. Basically, there are pretty high cumulative incidence of cardiovascular, endocrine, hematologic, and respiratory IRAs. So we actually conducted 3 models with Cox proportional hazards model. So in model 1, we only adjust for the age, gender, and race. And then in model 2, we also adjust for treatment type and initiation. Basically, we look at if the patient is put on combined therapy or PD-L1 therapy only. And thirdly, we adjust for the year of initiation. And in model 3, in addition to the variables that we adjusted in model 2, we also consider the duration of previous chemotherapy. And all 3 models are stratified by cancer diagnosis and baseline, because we want to allow a different baseline hazard for different type of cancers. So across the board, we sort of observe 5% lower risk of experiencing any IRAs in the treatment, the trial ineligible groups versus the trial eligible groups. So that moving on to, so I will explain a little bit why we observe this. So the key findings are the cumulative incidence of IRAs is consistent with observational studies that are conducted in other settings. So also with previous publications within the same population. And secondly, the real world participants treated with immune checkpoint inhibitors who would have met the hypothetical exclusion criteria had 5% lower risk of experiencing any IRAs compared to patients who did not meet the hypothetical trial exclusion criteria. So that's something that's quite counterintuitive in the first place, but usually when we understand IRAs, they're sort of reflective of patient immune responses. For example, it's possible that the patients with less ideal global function will have a weaker response to the immunotherapy, so that makes sense in a way. And also our definition of trial eligibility basically consider the patients using corticosteroids at baseline. So if they use corticosteroids at baseline, that means that it would reduce the chance of IRAs. So that might explain some of the findings that we observed. So the conclusion is that we should be cautious to interpret it as the patients with baseline autoimmune diseases or other conditions that are more likely to protect it from IRA. Rather, it might suggest that pre-existing conditions and concomitant treatments may make the risk of IRAs appear to be lower. So for a third aim, which is a comparative safety study in PD-1 users versus PD-L1 users. So why we conduct comparative studies in those users? Because PD-1 and PD-L1 inhibitors are usually considered interchangeable when they are prescribed for the same indication. So in RCTs, the efficacy of PD-L1 and PD-1 are pretty much comparable. So also we conducted in this aim, we also conducted a retrospective cohort study, very similar to the previous one. But here we have exposure that is PD-1 versus PD-L1 inhibitors. So the key point of this study is to really understand the causal effect of PD-1 and PD-L1 relative to each other in terms of the IRA experience. So we decided that we want to use propensity score and inverse probability weighting to make those two comparison groups more similar, so that we are able to derive causal relationships that's similar to clinical trials. So there are 4 types of propensity score usage, but here we use inverse probability weighting because it estimates the average treatment effect, and also it allows us to basically use a stabilized trimmed or trimmed weights to generate more stabilized results. So the specific steps that we took was, first of all, we fit a logistic model to estimate the propensity score. The variables that are in the propensity score model are the demographics and also some clinical characteristics that we mentioned before. And also we have some variables that reflects healthcare utilization, such as how many times the patients actually visited ER last year, etc. So, and then we applied a weight stabilization using the demographics again. And then we obtained the IPW, which is the inverse probability weights. And we use the weights to adjust the Kaplan-Meier curves and log-reg test to test the difference between the Kaplan-Meier curves. And finally, we use the adjusted Cox proportional Haddad's model to understand the relative effect. So for baseline characteristics, you might notice that based on the different agents that the patients initiated, actually in the baseline, they were very similar. They didn't differ much in age. They're pretty much the same in terms of sex distribution. And also there are very little difference in the race distribution, as well as recent brain metastasis, or corticosteroids use, or if they have any autoimmune diseases at baseline. So they're pretty much similar. And also, again, we look at the cumulative incidence by treatment, and we notice that basically they have very similar profile of safety as compared to what we observed in AIM-2. But here we notice that for hematologic adverse events and renal adverse events, they tend to differ in these two groups. So that's, so we did a analysis. We used the adjusted Cox proportional model, Cox proportional hazard models. So we, at the bottom of the graph, actually the relative hazard or the relative risk, the relative hazard ratio of any IRAs are not significant. They have very similar profile of experiencing any IRA, but they do differ in terms of the renal and hematologic IRAs. So the key finding in this aim is that compared to receiving PD-1 inhibitors, receiving PD-L1 inhibitors actually would increase the risk of experiencing hematologic IRAs by 14%, but would lower the risk of renal IRAs by 21.7%. So the implications are the oncologist might be able to choose a drug that meets individual needs. In this case, in our example, a lower risk of renal IRAs may justify that PD-L1 inhibitors have been largely used in urothelial cancer patients. And secondly, understanding of the risk of IRAs can provide insights on IRA management, including the magnitude and timing of IRA monitoring, treatment, suspension, and initiation of immunosuppressive therapies. So, for example, high risk of hematologic IRAs related to PD-L1 patients, PD-L1 inhibitor use may suggest closer monitoring on blood tests. So, in summary, here are some significance that stakeholders of these studies might be interested in. So for patients, the study definitely helped improve patients' outcome by understanding the nature of adverse events in immune checkpoint inhibitors therapies, and help patients with informed decision making on cancer management. So for providers, it improved the providers in the understanding of our ICIs with regard to adverse events. And for payers, definitely the utilization of the of ICRs has have been described. And for regulators, we have so the regulators might consider incorporate real-world evidence in their future decision making, and for trialists, and actually help them to think about how to improve the representativeness of trial population. So lastly, the limitations for the data, we exclude individuals with Medicare, Medicaid, or other fee-for-service Medicare beneficiaries, and the findings might not be generalizable to these populations. And we only capture patients who are treated with ICI. We miss the populations who are not treated with ICIs. So there might be a selection bias. For measurement, which is information bias, so some of the measurements are not perfect. For example, the algorithm that we use to predict ECOG performance status has a sensitivity of and a specificity of both at about 75%. And we also conservatively selected 4 criteria to evaluate the hypothetical eligibility. So there, in real world, patients might be disqualified from trials for many other reasons, in addition to the 4 criteria that we selected for our study. And lastly, the severity of the IRAs were not captured in our analysis. It's not even feasible to capture the severity in our analysis, but the severity might have some information about the overall response of immune checkpoint therapies, as well as survival outcomes. So lastly, in terms of a methodology, we used propensity score in AIM3, and the average treatment effect does not account for any individual effect. So it might be, so if individual want to get information from the populational effect, then it's probably not feasible. And lastly, the propensity score analysis require that all confounders to be measured and to be accounted for. We might have missed some, but we have already picked a set that's reasonably applicable in our analysis. So that's the end of my presentation. I know that's it's past the 1:15 time, but I will be taking questions.
Well, that is a lot of information to absorb, and nicely presented, though. So I appreciate that. I was curious, what was the rationale for excluding people on steroids in the clinical trials?
Yeah. So if this question is for trial eligibility, I'm not sure, but usually, I think in clinical setting, if you're taking corticosteroids, that means that you probably have some baseline autoimmune diseases. And also, for treating brain metastasis, sometimes you have to be put on corticosteroids. That's most often, corticosteroids is a type of chemotherapy agents that are used in combination with other chemotherapy agents. So these criteria are really sort of dependent on each other, because to be qualified for trials, you can't have corticosteroids corticosteroids use, you can have autoimmune diseases, you can have recent brain metastasis, you also can have recent chemotherapy. So those are basically very dependent on each other. So I would say that so corticosteroids is so one of such criteria.
So this is Doug. The other issue is corticosteroids. Remember, you're inducing an immune response to the tumor.
Yes.
You're allowing the immune response to occur. Corticosteroids suppress the immune response. So if you're on corticosteroids, you may be reducing the effectiveness of the drug. And at least in the clinical trial, you don't want to have patients where they'll have reduced effectiveness.
Yeah, so that's also why in M2 we observe that there's a lower risk of immune-related adverse events in those patients who are not qualified for trials because they are on corticosteroids.
And I'm not sure if I'm remembering correctly, but I think in the checkpoint studies, the corticosteroids were the rescue mechanism if adverse effects occur. So maybe that is also a point.
Yes, correct. So we actually, I was, so before this, I actually was proposing that if we can look at post-ICI initiation corticosteroids use, because that means that patients are treated for immune-related adverse events. But it's quite unfeasible because usually the corticosteroids are initiated before the adverse events actually happen. So in the claims database, it's quite difficult to capture that as well. So we decide not to do that, and instead we want to characterize, what's the proportion of corticosteroids used at baseline.
I have a question. I was wondering, maybe I missed it, but what I have learned is that the most common effects are on the skin and also gastrointestinal adverse effects. And when you had your table, I think for the most common, I think you didn't list them. Then when you showed the hazard ratio, I saw the gastrointestinal, but normally I think rash was quite high on the frequency.
Yeah, that's a very good point. I didn't include skin adverse events because usually they're described in their EHR rather than put in the diagnosis code. That's a very good point because the most common skin adverse events is just rash on the skin. So it's very difficult to be captured. And secondly, in regards to the gastrointestinal adverse events, yes, there are a lot, especially in diarrhea or colitis. But the problem is that the diagnosis for colitis is not that many. Rather, diarrhea might be captured in EHR data just by free tests in the medical record. But also there's one thing I forgot to mention is that in AIM-3 we're only looking at PD-L1 patients where there is a different spectrum of adverse events. Usually we see a lot of diarrhea and colitis in CTLA-4 inhibitor users, but not that many in PD-L1 users. But PD-L1 users might have higher endocrine adverse events or other adverse events. So those two agents do have a different spectrum of adverse events. So that's why we're not seeing that many gastrointestinal adverse events in our case.
In your definition of trials, you said that they had to be on the label. Don't you think there's trials that don't get on the label that are maybe part of the process?
There are a lot of trials that are not in the label or they're in the process of new indications. But I think the point to use those published trials that are in the label is that we want to, so because the evidence that we have seen, either for efficacy or for adverse events, are generated from those trials that are pivotal. So I think that's why I only use those trials that are in the label rather than have a broader set of trials. Not sure if I understand, if I answered your question.
Yes, and I'll have a couple opportunities to ask again. I think it looks like there are no other questions. If I'm
okay. Any other comments or?
Okay, well, thank you, Jiajun. I'm going to take over for Mark. But nice job, and it looks like you're progressing quickly towards defense.
Yeah, thank you.
Okay, thanks everyone for attending.
Thank you all.
Thank you very much. Bye-bye.
Bye-bye.